Here are the goals for the second half of this class. We're going to finish up the visual system. We're not going to get to, perception, but we are going to get to vision. So we've already done the first bullet, understand the molecular mechanisms of phototransduction. You have that now, and I want you to understand it. And feel free to ask me questions. Oh, and by the way, let me make an announcement that the lab is open today until midnight and tomorrow until midnight, but it won't do you any good after 9 p.m. But you can come into the lab and use AMP Revealed if you want to use that program to help you review the muscles that are on the quiz and the brain structures that are on the quiz. So the lab is open today and tomorrow. All right, so we've done bullet number 1 here. Now we're going to get into, well, we've kind of already done this too. Lateral inhibition by horizontal cells and amacrine cells to focus the response. And then we're going to get into the other cells of the retina and what they do. So I'm not going to tell the story again. I feel like I've told the story just recently, a few minutes ago. But you should be able to narrate this story. And you should be able to narrate it blind, without seeing anything. Right? You should be able to narrate it in your sleep, in the dark. Okay? You should understand why the resting membrane, the potential of photoreceptor cells in the dark, at rest, is relatively depolarized, and how the molecular mechanisms for graded hyperpolarizations due to stimulus, light. So you know how we turn light into a hyperpolarization of the first cell, the photoreceptor cell. And you already know that that causes a decrease in neurotransmitter release. Now let's jump to the other end, right? The output along the optic nerve. Let's just jump to see that's where we're starting. This is where we're starting. Now let's see where we're going. We're going to get to ganglion cells that have different responses to light. So there are ganglion cells. So what is this supposed to show? This is supposed to show a spot on a retina. The center of so ganglion cells in here, ganglion cells are out here. A spot of light shined on these photoreceptor cells and not on these photoreceptor cells. Let's not worry about a spot of light in the periphery. Let's not worry about that. Let's think about a spot of light on the center of a response area. Now here's the key. There are ganglion cells whose frequency of action potentials goes up when light is shined on the center of that area. These are the so-called on-center cells. Let's not worry about the fact that they are also off-surround. They are on-center. On. Increase. So these blue bars are action potentials on a very compressed time scale. You understand them: sodium channels, voltage-gated sodium channels, voltage-gated potassium channels, etc. When light shines on the photoreceptor cells that are connected to these ganglion cells indirectly, the frequency of action potentials of these ganglion cells, axons, go up. There are also ganglion cells where light on the center causes a decrease in action potential. Light turns off action potential frequency, and these are the so-called off-center. Let's not worry about the surround. Okay? So if we were to, if we imagine vision, light hitting our retina, hyperpolarizing photoreceptor cells. Now there's going to be some things that I'm going to unpack in just a minute. And we jump to the output, the action potentials in the axons of the optic nerve. Some of those axons are carrying more action potentials, and some of those axons are carrying fewer action potentials. And the pattern of action potentials, high frequency or low frequency, is what our brain is going to interpret as image. The point here is that both an increase in frequency and a decrease in frequency is information. Now what I want to do is unpack how a hyperpolarization of the photoreceptor cells is converted into either an increase in action potential frequency in the output or a decrease in action potential frequency in the output. That's where we're going. How does that work? It works like this. I'm going to go. So you only have one slide because this is one of those slides that builds. So the slide in your handout is very hard to understand. The slide on Moodle is going to build the way I'm going to build it. So my recommendation, my strong recommendation is when you're studying this and trying to reinforce your knowledge of what I'm going to teach you now, you don't use the static handout. You go to Moodle, you pull this up, and you build it with me. Okay, so let's build this. Here's a photoreceptor cell. It is connected to two different types of bipolar cells. One photoreceptor cell, two different types of bipolar cell, two different types of ganglion cell. You can almost already see where I'm going. Two different types of ganglion cell. One of them is going to have a higher frequency of action potentials, and one of them is going to have a lower frequency of action potentials when light hits this spot. Right? This is the spot that is connected to two ganglion cells in their center. And one of them is going to increase the action potential frequency, and one of them is going to decrease. Okay, so let's build this. Brittany, can you dim the lights? Because I think the color will matter. Okay. Everybody with me? Photoreceptor cell, neurotransmitter release, bipolar cell, neurotransmitter release, ganglion cell, action potentials. Let me tell you the commonalities here. Photoreceptor cells hyperpolarize in response to light. It decreases the amount of neurotransmitter release. This neurotransmitter is glutamate. So that's a thing to know. This neurotransmitter is glutamate, and this neurotransmitter is glutamate. So glutamate is the neurotransmitter of the synaptic processing of light input. But how do we get high and low action potential output? Okay, light. Light hits the photoreceptor cell. We're in the center of a field. Okay, that was just an electrode. Here's the output from the electrode. The yellow bar represents the period during which light is on. What happens to so this is voltage as a function of time. Voltage as a function of time. What happens, because we're measuring voltage with this electrode, what is this image going to look like? What happens to voltage when light hits it, the yellow bar? It hyperpolarizes. There's the response of the photoreceptor cell again. You should understand the molecular mechanisms for that. That was in the previous slide. What does this hyperpolarization do to the calcium channels here? Allows them to close. What does that do to the amount of glutamate that is released here? Decreases it. So, decreased glutamate release. Now here at this, so if there's one thing you have to understand, it's this synapse. On some of these bipolar cells, there are ionotropic glutamate receptors that are sodium and sometimes calcium permeable. You already know what those are. Those are excitatory glutamate receptors. So a decrease of glutamate causes decreased excitation. That's easy. On other bipolar cells, there is an inhibitory metabotropic glutamate receptor. This is a GPCR that is coupled to GI. So, and it's inhibitory. So tell me, you know, what happens when I decrease glutamate in this synapse right here? Well, I'll tell you. Decreased activation of an inhibitory receptor, decreased inhibition is allowing these cells to depolarize. So this is decreased excitation causing hyperpolarization, and this is decreased inhibition causing depolarization. The decrease at the very beginning was the decrease in glutamate. Decreased glutamate, decreased excitation, decreased neurotransmitter release because of hyperpolarization, decreased glutamate, decreased inhibition, inhibition of inhibition, depolarization, more glutamate release. Okay, let's just see we put some electrodes in here. I forget what the next build is. Light is going to cause, so this one is decreased inhibition. Light is going to cause a depolarization of the dark blue. Put another electrode in here over in the light blue. Light, decreased glutamate release, decreased excitation is going to cause a hyperpolarization of the light blue. You can see why the black and white static thing gets very hard to interpret here, but it becomes pretty clear if you walk through these step by step, especially with color. So that was the key synapse. Because now you can see, okay, decreased glutamate release, decreased excitation of this cell, decreased depolarization. The yellow cells are going to be the off-center cells. And the orange cells are going to be the on-center because we're going to have more glutamate, more depolarization. Oh, what I didn't tell you is here at this synapse, we have excitatory glutamate receptors on both of them. So let's build it. We're going to record from this axon light, more glutamate, more excitation, excitatory glutamate receptors here, more excitation, more depolarization. More depolarization would lead to a higher frequency of action potentials, and the orange cell is the on-center cell. Let's do the other one. Recording action potentials from the other neuron, light, light, decreased excitation, decreased depolarization as a hyperpolarization, decreased glutamate, decreased excitation, hyperpolarization, fewer action potentials, onset. Offset. Light on, fewer action potentials.
Hey, Bob.
Yes.
So I'm confused about the dark purple bipolar cell.
This one.
So when light strikes the photoreceptor cell, there's decreased glutamate release, so decreased inhibition of the cell, but there's no excitatory signal, so it just, in the absence of inhibition, it will just spontaneously release transmitters?
No, well, yes and no. No. In the absence of inhibition, there's enough leakiness of this cell to allow a depolarization. So less inhibition, leakiness, depolarization, more voltage-gated calcium channels open, more calcium influx, more glutamate release. So the thing that was missing was understanding that if you relieve this cell of its inhibition by inhibiting inhibition, there's enough basal leakiness to allow depolarization.
Okay, cool. Thank you.
That was a good question, because I sort of live in a world where I think of leakiness, but you have to remember that. Other questions? So my recommendation is walk through the story as many times as it takes to get it. What you'll notice is that expressing the inhibitory receptor here, so inhibition of inhibition, is the thing that leads to on-center. And inhibition of excitation is what leads to off-center. I do not want you to memorize this string of characters. I do not want you to memorize this string of characters. You don't need to know that. What you do need to know is that some of the bipolar cells express an excitatory glutamate receptor, and some of the bipolar cells express an inhibitory glutamate receptor. And in both cases, light causes an inhibition of glutamate release. Okay, so now we have action potentials, either increased or decreasing frequency. You can go ahead and turn on the lights now, I think. We have action potentials, increased or decreased frequency in the optic nerve, in axons of the optic nerve. Now we need to take that information to the brain. There's an interesting thing that happens here in the optic chiasm. Watch what happens here. So in the optic chiasm, some of the axons from both eyes cross over. Which ones cross over? It's the ones in the medial part of the retina. So here's the fovea, sort of in the middle. Fovea, medial retina, lateral retina. Fovea, medial retina, lateral retina. The medial retina crosses over. The axons from the ganglion cells in the medial retina cross over. But the axons from the lateral retina, the axons from the ganglion cells in the lateral retina do not cross over. What this means, functionally, is that both, so we're going to the lateral geniculate nucleus of the thalamus. That's where we're going. Both lateral geniculate nuclei get information from both eyes, right? We get information from the left eye to the left lateral geniculate nucleus, and we get information from the right eye to the left lateral geniculate nucleus, and vice versa. So what does not happen is that left eye goes to the right side and right eye goes to the left side. That is not what happens. What happens is that the left visual field, so now I'm putting my fovea on the camera. I've got a left visual field and I've got a right visual field, right? Looking at the camera. Left visual field, right visual field. Information from my left visual field is going to the right side of my brain. Information from my right visual field is going to the left side of my brain. Left visual field, right thalamus, right lateral geniculate nucleus of the thalamus. Right visual field, left lateral geniculate nucleus of the thalamus. Here's how that works. So we're looking at it from above, or from below, doesn't matter. We've got a left field and a right field, so I'm looking at the camera. Both of my eyes are seeing my left field. The left field is projected onto the medial part of the retina of the left eye, and the left visual field is projected onto the lateral retina of my right eye because of optics. Well, the medial part of my left eye and the lateral part of my right eye are both going to my right lateral geniculate nucleus. So the left visual field, because of optics and because of selective crossover at the optic chiasm, take the information to my contralateral lateral geniculate nucleus, contralateral to the field, not contralateral to the eye. So my recommendation is to be able to reproduce this image from memory. If you can reproduce this image from memory, you can figure out a problem that I might challenge you with in terms of which thalamus is getting the information, which lateral geniculate nucleus. Okay? So unlike the previous one, this image is pretty good in a static mode. I do not recommend trying to memorize this image by itself because it's not informative by itself. Build it. But this one you can just learn and understand. Understand what's happening at the optic chiasm. Is there synapses going on here? Are we talking about other cells? No, there's no synapses. These are axons going, and the next synapse is in the thalamus. So let's go back and talk about synapses for just a minute. First synapse of the visual system is in the retina. Second synapse of the visual system is in the retina. Third synapse of the visual system is in the lateral geniculate nucleus of the thalamus. Which thalamus? Which side depends on where the image is. In the fovea, we get a mixture. This is just another image. I'll skip this. So the lateral geniculate nucleus receives information from both eyes, right? The lateral geniculate nucleus receives information from the contralateral field, but it receives information from both the contralateral. So we're talking about my left lateral geniculate nucleus. I'm getting information from my right eye and my left eye, but there's segregation of that input. And do not memorize these numbers. There are different layers in the lateral geniculate nucleus, and the different layers of the lateral geniculate nucleus receive information from the different eyes. So if I'm talking about my left lateral geniculate nucleus, I'm receiving some information from my left eye and some information from my right eye. And it's segregated into different layers, but don't memorize which layers are which. I might on a problem set ask you to solve a problem with this, but that's open book. You can learn it. I mean, you can look it up. What does the thalamus do with this? The thalamus is a relay and processing center. So it's going to relay the information to my visual cortex, but it's not just a simple relay, it's a processing relay. So some of the cells in the thalamus are sensitive to motion, and some of them are specialized for sharpness and color. There's separation, so in the thalamus, we start to separate processing of an image that is moving. So this is kind of interesting. If I put this pen in my pretty far in my peripheral vision, so I'm staring at the camera, I know there's a pen here because I know I'm holding it, but I can barely see it. It's really hard to see. But if I move it around, I know where it is because I'm using my motion sensitive cells of my thalamus. If I put this pen in my fovea, I can make sharp color information. Why do we care about movement in our peripheral vision? Well, you know, if there's something coming at us, like a baseball coming at me this way, you know, I'm going to respond quickly, get out of the way of that baseball. Or, you know, if it's a dog running at me, I was just like, okay, what, is that a friendly dog? Or, you know, then I start to use my cognitive skills to determine whether it's dangerous or not. But we're good at detecting movement in our peripheral vision, both because that's where the rods are focused and because there are cells in the thalamus that are paying attention to that and good at processing that information. So questions about what the thalamus, the lateral geniculate nucleus of the thalamus is doing. It's receiving information from both eyes. It's receiving information from the contralateral field of both eyes and processing it. And sending that information to the visual cortex. The primary visual cortex in the occipital lobe. This slide also shows some interesting things that I want to mention, but I will not examine you on it, but I might ask you to use it in a problem set context where you can look it up. Optic nerve. Crossing over in the optic hiatus. This is one that's crossing over. Here's the lateral geniculate nucleus. This is what's important for seeing. But you know we do other things with light. So some of that information goes to the hypothalamus and starts to regulate our day-night cycle, right? Where we tend to be awake at night. No, we tend to be awake during the day, and we tend to be asleep at night. Generally, most of you violate that to some extent, but we evolved to be awake during the light and to be asleep during the dark. So when you stay up till 3 o'clock in the morning, you are violating your evolutionary principles, the same. We evolved to be awake during the day, and the hypothalamus helps control our day-night cycle, and it is receiving information from the eyes so that it knows, is it light out or is it dark out? There is an area of the brain called the tectum, the pretectum, that controls the output of the oculomotor nerve to the iris that controls the pupil. So it says, am I getting a lot of light? I need to constrict the pupil. Am I getting not very much light? I need to relax the pupillary sphincter and dilate the pupil. There's a part of the brain that does that. That's not, that decision to constrict or to dilate is not done in the retina. It's done in the tectum, part of the brain. And there's a part of the brain that receives information that helps me coordinate my head movement, head and eye movement. We've learned, so I'm focusing on the camera, and now I'm focusing on that house across National Road. I'm moving my focus from here to there. I can do it without moving my head, or I can do it with moving my head. And I'm pretty good at both of those. Because I've learned, using my superior colliculus, how to do that. Some of you may have, if some of you have ever looked at the eyes, into the eyes of a newborn baby, their eyes are wobbling all over the place. Really, it's kind of creepy looking. They're not forming. They haven't learned yet how to move their eyes together. Their eyes are wobbling around, independent of each other, and they're not focusing because they haven't learned how to focus. They haven't learned how to coordinate their eye movement. They look weird because this is a learned process. We learn how to control our head and our eyes so that we can focus on something, quickly and move our head and focus on something else. That's in the superior colliculus. The lateral geniculate nucleus is the part that's important for vision. But we also use light to control our diurnal cycle, to control the pupillary diameter, and to control balance and movement. Now let's go back to vision. What happens here in the visual cortex, the primary visual cortex? Primary visual cortex is in the posterior occipital lobe, above and below the calcarine sulcus. I do want you to know the name of the calcarine sulcus and posterior occipital lobe. That's part of the neuroanatomy of vision. Calcarine sulcus or calcarine fissure. There's a gyrus above it, and there's a gyrus below it. They have names that I'm not going to ask you to know. So I do want you to know the name of the sulcus, but I do not care that you learn the name of this gyrus or this gyrus. I'm not even going to say it so that you have to look it up if you want to know. Now, just to be clear, I could ask you the name of this, or this, or this, or this. I could ask you the name of this, this, all, here's a good slide to test yourself on your neuroanatomy in a midline sagittal section. I am not on the quiz going to ask you about vision, but there's lots of stuff on here that I could ask you about. Primary visual cortex. There is what's called a retinotopic map. There's a map of the retina on the primary visual cortex. So let's look at this. So here's the fovea. Here is the left visual field of the fovea and the right visual field. So fovea, so this is all looking at it's the left visual field, including the fovea. So it's from here over. The fovea of my left visual field, remember the left visual field ended up, the information from my left visual field ended up in my right lateral geniculate nucleus, and it ends up in the right primary visual cortex. So left visual field, the fovea, is most posterior, is mapped most posterior. So just like there is a map of the body on the somatosensory cortex, right, with the face lateral and with the legs medial, etc. There's a map of the body on the somatosensory cortex. There's a map of the retina on the visual cortex. The fovea is posterior. The periphery is less posterior. The lower visual field, 2 and 4, is mapped to the gyrus above the calcarine sulcus, and the upper visual field, 1 and 3, is mapped to the gyrus below the calcarine sulcus. So this is a crude map of the retina, of the left visual field, on the right cortex, fovea, posterior, periphery, less posterior. Upper half to the gyrus below the calcarine sulcus. Lower visual field to the gyrus above the calcarine sulcus. I do want you to know this general map. So now we're getting very close to perception, which is not where we're going to go with this course. But I do want you to know that there are two visual streams from the primary visual cortex to other parts of the brain. Right? So the primary visual cortex is not all we need for vision. Remember, vision is complex. It's a construct of our expectations. It's a construct of what we know we're going to see. It's not just happening in the primary cortex, primary visual cortex. So there is a stream, that is to say, axons carrying information from the cells of the primary visual cortex to the parietal lobe that is primarily interested in where, the location of whatever it is I'm thinking about or seeing. Where is it? The dorsal stream is carrying where information. The ventral stream from the occipital lobe, the posterior occipital lobe, to the temporal lobe is a ventral stream of information, and is carrying information about what is it. So, is it a dog? Or is it a bear? Is it a friendly dog, or is it a friendly bear? Is it a Nerf ball, or is it a baseball? Those are important information about what it is, and then is it coming at me, or is it running away? Is it coming straight to me, or is it going to miss me? So those are the streams of visual information. And of course, we process this in a very complex way in order to form vision and in order to inform our responses to visual input. So what did we just do in the past 2 hours? We went from a photon hitting our retina, and a lot of information about that, to information streams in our brain. So we covered a lot of ground in a not very long period of time. I hope I have made it clear what I want you to know and what I was just talking about because I think it's interesting. I've tried to reduce the latter and I've tried to increase the former. I've been putting my brakes on certain interesting things. But I welcome your questions. Remember, this is not on the quiz 2, but it will be on problem set 2, 3, and problem set. It will be on problem set 3 and quiz 3. And those are not very far away. So let me end by reminding you that quiz 2 is due tomorrow. Problem set 3 is due on Friday. And I'll make that available as soon as we're done with class today. And quiz 3 is due on Sunday. So I'm not pushing things back anymore because we have an exam a week from Wednesday. Okay, so on Wednesday, this Wednesday, we're gonna cover auditory, olfactory, and taste. So I'm gonna use the full time. And then on Monday, we're gonna do motor. And then on Wednesday, we got a test. So we're gonna be moving pretty fast here. Remember, you need to work on this at home in order to get it and understand it. I welcome your questions. Brittany welcomes questions or tutoring sessions. But that's where we're going. So don't let quiz 2, which is due tomorrow, cause you to forget about the things that are coming up pretty soon after that. We're moving along. But we're done for today. Enjoy the rest of your day.